Hi, I will present a research project that's still under development led by André Luiz Acosta called Coronavirus from Cities to Forests, Mapping Vulnerable Interfaces and Hotspots for SARS-CoV-2 Spillover from Humans to Biodiversity. Firstly, we need to highlight a basic concept. Zoonotic disease spillover occurs when a pathogen jumps from one infected animal species to another, eventually spreading into their population. This is a common process in nature. It's estimated that more than 70% of human viral diseases is originated by viral spillover from other animals. For our analysis, we have to assume the origin of SARS-CoV-2 probably came from a three-step process, with the emergence in bats and jumping to pangolin and then jumping to humans. But we are now aware of the vulnerability of several species to contract SARS-CoV-2, including cats, dogs, monkeys, ferrets, lions, tigers, camels, civets, and other domestic and wild animals. Here in Brazil, the interaction between humans and monkeys is very close and frequent, and monkeys have contact with contaminated waste with human blood, feces, and fluids, severely increasing the risk of a human-to-animal spillover event. Here we also have bats everywhere, in ceilings of houses, in food stores, animal breeding places as cattle shed, chicken farms, and pigsties. And of the three hematophagous bat species on the planet, Brazil has all of them. Considering the susceptibility of species and their close interaction with humans, we infer that species from these orders are highly vulnerable to a human-to-animal spillover. The most probable scenario is the spillover from humans to primates living at the edges of reserves and urban parks. Once one monkey becomes infected, it can take the virus to primates populations and other species in the forest's core. Likewise, bats that transit between urban and wild zones would take the virus to their populations. Since bat species shares resources among species, new spillover events would take place into the forest. Monkeys and bats share food and shelter resources in tree canopies and cavities, increasing even more the risk of spillover. Aiming to map zones of highest species richness, we obtained bats and primates distribution data and modeled their habitat suitability by using BioMod2. Subsequently, we combined the results generating maps of potential species richness per order. After that, we crossed with land use, forecast cover, and human demographic data in order to detect places where we can find a higher amount of vulnerable species in forest edges under human impact, and also near zones with high human density. These preliminary maps show hotspots most likely for spillover events in Brazil from COVID-19 infected humans and wild species. These areas can be priority zones for fauna sampling and monitoring to detect SARS-CoV-2 or new variants. We are improving our methods aiming to obtain maps with highest accuracy possible. Thank you for your attention.